Yes, sir.
ಮ್ಯೂಟ್ ಈಗ ಆಯ್ತು. ಅವಾಗಿಂದ ನಾನು ಕಾಯ್ತಾನೇ ಇದ್ದೆ ನಾನು. ಓಕೆ. Good morning, students. In today's class, we'll be studying about reproduction in organisms, and we'll be solving a few MCQs in reproduction organisms, and then we would study microbes in human welfare and some MCQs in microbes in human welfare. Prajwal sir, can I start?
Sir, ಹೇಳಿ ಸರ್.
Start ಮಾಡ್ಬೋದಲ್ಲ?
ಮಾಡಿ ಸರ್.
One second.
ಡನ್ ಸರ್, ಶುರು ಮಾಡಿ.
ಡನ್ ಸರ್. ಓಕೆ ಸರ್, ಓಕೆ. ಈಗ ಶೇರ್ ಸ್ಕ್ರೀನ್ ಮಾಡ್ತೀನಿ. ಒಂದು ನಿಮಿಷ ಸರ್. Good morning, students. Now we'll start studying about reproduction in organisms. So what is reproduction? Reproduction is a fundamental feature of living organisms which involves transmission of genetic material from one generation to the next, ensuring the survival of the species. Along with that, there is transfer of genetic material. This is very important for the survival process of species over a long period of time. So in very simple terms, we usually use the word offsprings, and we say reproduction is a technique where, or a phenomena where adults will produce offspring. So one species will produce those offsprings for the propagation of its species. Then here we have a definition called as lifespan. What is lifespan? The period from birth to natural death of an organism is called its lifespan. So lifespan of an organism may be few minutes to a several thousand years. See, we have different organisms here. Please do remember their lifespan. So one of the most important would be the parrot. Even though it's a small organism, so it lives for nearly 140 years, compared to a dog which is much more bigger than it, which lives only for 25 years. Okay. Elephant 60 to 75 years, horse around 50 years, whereas a tortoise, which is even more smaller, will live to 100 to 150 years. Please do remember all the lifespans of different organisms, which is very important. Please do remember these are important. Then we go to the next part, phases of life. Lifespan of an organism includes 4 phases, that is the juvenile phase, a reproductive or maturity phase, aging or senescent phase. Later after that is death. So what happens in the juvenile phase? In the juvenile phase, the organism is just born and will undergo growth. So one second, it will undergo growth, where it grows vegetatively. All the reproductive structures are not developed during the juvenile phase, whereas in the reproductive phase, the organism will reach a condition with puberty and then reaches the reproductive phase. In the reproductive phase, all the gonads and reproductive structures are well developed and the organism will be able to reproduce. Once the reproductive phase or the maturity phase is done, it enters the aging phase or the senescent phase. In the aging phase or the senescent phase, what happens is the organism's reproductive capacity is ceased and it wouldn't be able to reproduce, and the cells start degenerating and the tissues start degenerating, and it would be leading towards death. So remember this. Then, which are the different types of reproduction? The different types of reproduction are asexual reproduction and sexual reproduction. In asexual reproduction, we have different types. So before that, let's see what is asexual reproduction. Asexual reproduction is a type of reproduction where there is no involvement of gametes. It is uniparental. Large number of offsprings are produced. At a single reproductive phase. Okay, large number of offsprings when compared to reproductive cycle. Then sexual reproduction, and the offsprings that are formed are called as clones. Then let us look at the different types: fission, fragmentation, regeneration, budding, and vegetative propagation. These are the different types of asexual reproduction. So in fission, we have 2 types. One is binary fission, the other one is multiple fission. Binary fission, the cell divides into 2, whereas in multiple fission, the cell divides into many smaller cells. This is multiple fission. Okay. So in fragmentation, the individual plant body would get fragmented, or it would get, the small part would get pinched off from the main part, and it would form a new organism altogether. In regeneration, the lost parts are regenerated. In budding, a small bud, it might be exogenous or endogenous, which are produced, which will pinch off. Usually they are produced in some organisms in unfavorable conditions. Then we have vegetative propagation. So what is vegetative propagation? Where the vegetative parts of the plant, will give rise to new plants. So one of the most common vegetative reproductive structures, the first one, runner. So runner is seen in mint. Can you see mint? A rhizome in ginger, leaf buds in bryophyllum, tuber in potato, offset in iconia, and bulb in agave. See, here you need not remember the definitions here, because in NEET they wouldn't ask for a definition much, but the examples are very, very important. Then coming to the next part, asexual reproductive structures. Look at this. Zoospores produced in Chlamydomonas, which are biflagellate motile spores. Conidia produced in Penicillium. Buds in Hydra, gemmules in sponges. Then coming to sexual reproduction. In sexual reproduction, it involves the formation of male and female gametes. It is either uniparental or biparental, or by different individuals of the opposite sex. So it can be both uniparental or it can be biparental. Uniparental or biparental. In uniparental, only one individual is involved. In biparental, two individuals are involved, where one is considered as the male and the other as the female. So remember this is very important. Then the gametes fuse to form zygote. The zygote is diploid. The gametes are haploid, which develops to form a new organisms. Then looking at plants. So there are different types of plants. The first one is annuals. The plants which complete its life cycle within 1 year is called as annuals. Example is pea. Remember this, very important. Then plant which completes its biological cycle in 2 years is biennial, that is onion. Perennial, the plant that completes the plant that lives for many years, that is more than 2 years, is called as perennial plant. Classical example is mango. Here in perennial plants, you can have plants which flower throughout the season, okay, throughout a particular season, and they don't flower for the rest of the year. They are called as seasonal plants. Whereas some plants flower throughout the year in all season, okay, in all the seasons. Okay, they are called as non-seasonal plants. For example, mango is a seasonal plant, so it gives flowers and fruits only in a particular season. Okay, whereas coconut, some flowering plants, they flower throughout the year. Okay, then some plants are unusual. Some plants are very unusual in their phenomena of flowering. Bamboo. So bamboo species flowers only once in their lifetime. Understand this, bamboo will flower only once in a lifetime. Generally after 50 to 100 years, they produce large number of fruits and they die. Then you have Strobilanthus kunthiana. Also called as Neela Kurunji. This Neela Kurunji flowers only once in 12 years. Are you understanding? This will flower only once in 12 years. The plants are flowered during September to October in 2006. Now in 2018 they have flowered. The next flower will be in 2030. So do remember this. They flower only once in 12 years. Then coming to breeding of animals. So on the basis of breeding, animals can be considered into two groups. Okay, one is seasonal breeders, the other one is continuous breeders. Seasonal breeders, they reproduce at a particular period of the year in a particular season, such as frogs, lizards, and most birds, deer, etc. Whereas continuous breeders, The animals continue to breed throughout their sexual maturity. Example, honeybee. Okay. Then poultry, all this chicken, rabbit, mice, cattle, etc. Cattle, etc. They breed throughout the year. Then, what are the reproductive changes during reproduction? So, what are the changes that you see during reproduction? So, there are many cyclical changes that we see during reproduction. One of them is estrus cycle, and the other one is menstrual cycle. In estrus cycle, the cycle takes place in non-primate mammals, that is cow, sheep, dog, and tiger. So here in menstrual cycle, it takes place in primate mammal, example monkey, apes, and humans. In menstrual cycle, there is shedding of exoskeleton, shedding of endometrial wall, there is shedding of endometrial wall, which leads to bleeding in a particular phase of the cycle. So this is very important, which does not take place in estrus cycle. Then what are the sexual reproduction events? One is pre-fertilization, fertilization, and post-fertilization. So pre-fertilization events are the events that takes place before fertilization. So what are they? Gametogenesis and gamete transfer. What is gametogenesis? The formation of gametes is called as gametogenesis. So what about gamete transfer? The transfer of gametes from the male individual to the female is called as gamete transfer. Okay. So which leads to fertilization later. So gametogenesis, you have to remember this. There is formation of male gamete and the female gamete. For example, in humans, it is called spermatogenesis and oogenesis. So you can have two types of gametes. One is homogametes or isogametes. Okay. What are homogametes? The two gametes are similar in appearance. Understand this? They are similar in appearance, can not categorize into male and female gametes. Example in cladophora. So they're absolutely same. You just can't, by looking at them, you can't differentiate them into male and female. What about heterogametes? The two morphologically distinct gametes. The two morphologically distinct gametes are called as heterogametes. So male can be called an anthrozoid or a sperm. Female can be called as a egg or ovum. Okay, and there is an other condition where the male gamete is small and motile. Okay, male gamete is small and motile, whereas the female gamete is large and immotile. So what is a large male large female gamete? It is stationary, whereas the small male gamete is motile, and it actually moves from the male individual to the female. Then we go to the next one. So sexuality, they can be homothallic and monoecious. Remember this? Means terms used to describe bisexual condition of a plant. That means they have both male and female structures. Heterothallic or dioecious, the terms used to describe unisexual condition of a plant. So unisexual conditions of a plant, that means both of them are present in different plants. So male is present on different plant, female is present on another plant. Then staminate, unisexual male flower bearing stamens, and pistillate, unisexual female flowers bearing stay. Okay, one second. Here it is carpels. Or pistils. Sorry, it is carpels or pistils. Then monoecious, both male and female flowers may be present on the same individual. Example, cucurbits and coconuts. Is sexual reproduction uniparental? Sexual reproduction can be both uniparental and biparental. So sometimes if it's a monoecious organism, so both the male and the female can be present on the same plant, okay, or the same individual, then it is uniparental. Okay, whereas asexual reproduction is absolutely uniparental. Okay, whereas sexual reproduction can be both uniparental and biparental. Okay, then so dioecious, both male and female flowers may be present on separate individuals. Example, papaya and date palm. Please remember these two examples, papaya and date palm. They are very important, papaya and date palm. So these are some of the sexually reproducing organisms. Then so look at this, cell division during gamete formation. The parent body of an organism like Monera, fungi, algae, and bryophytes is haploid. They produce gametes by mitotic division. They produce gametes by mitotic division. Then the parental body of an organism like pteridophytes, gymnosperms, and angiosperms. Most of the animals, including human beings, are diploid. They produce gametes by meiotic division. They produce gametes by meiotic division. So the gametes are haploid here. In diploid organisms, specialized cells called meiocytes, that is gamete mother cells, undergo meiosis. So remember this. Then gamete transfer. In majority of organisms, male gamete is motile, while the female gamete is stationary. So that is oogamy. Exceptions, in few fungi and algae, both the types of gametes are motile. The medium required through which the male gametes move is usually water. In algae, bryophytes, and pteridophytes, water is the medium for gamete transfer. Remember this: algae, bryophytes, pteridophytes. And also remember, few angiosperms are also aquatic plants. Example, in the case of Vallisneria, many hydrophytic plants, they are also angiosperms. So their medium of transfer is water only. To compensate the loss of male gametes during transport, the number of male gametes produced in several thousand times the number of female gametes produced. Why? Because of the flow of water, current that is there, and also because of movement of other organisms, the male gametes might be lost. So remember that usually male gametes are produced in large numbers when compared to female gametes. Okay. The next step we see, in plants, they undergo pollination. What is pollination? Transfer of pollen from anther to the stigma is called as pollination. Anther to stigma is called as pollination. Some plants undergo self-pollination and some of them undergo cross-pollination. What is self-pollination? Where the pollen grains of the plant will fall on the stigma of the same plant in on the same flower. That is very important. That is self-pollination. Whereas if the pollen from the anther fall on the stigma of some other flower, then it is cross-pollination. And even if it's of some other plant also, it is cross-pollination. Then pollen grains germinate on the stigma and the pollen tubes carrying the male gametes each to the ovule and discharge the male gametes near the stigma, near the ovary. Then they get fertilized. So remember this: pollen grain, pollen tube formation, fusion of male and female gamete, then fertilization and syngamy. So the last step, fertilization results in the formation of a diploid zygote. That is 2N, two sets of chromosomes. Then parthenogenesis. What is parthenogenesis? Some of the female gametes undergo development of new organisms without fertilization. This is called as parthenogenesis. Example, the classical example is honeybees. Remember this, honeybees. Okay. Then fertilization can be internal or external. If it is external fertilization, it takes place outside the body of the organisms. Example, algae, fishes, and amphibians. Whereas internal fertilization takes place inside the body of the organism. Example, fungi, reptiles, birds, and mammals. Then the post-fertilization events. Events in sexual reproduction after formation of zygote are called post-fertilization events, Then the formation of diploid zygote in organisms belonging to fungi and algae. The zygote develops a thick wall that is resistant to desiccation and damage. It undergoes a period of rest before germination. Understand this. Then the process of development of embryo from zygote is called embryogenesis. The zygote will undergo cell division, that is mitosis, and later cell differentiation and specialized tissues, then to organs and organ systems, and later the organisms. Then the animals can be of two types. One is oviparous, the other one is viviparous. Oviparous are organisms which lay fertilized or unfertilized eggs. Example, reptiles and birds. Viviparous are those organisms which lay, which give birth to young ones, which give birth to young ones. Then, if it is oviparous, usually it is covered over by a calcareous shell. Then that type of egg is called as a cleidoic egg. Cleidoic egg. In viviparous, the proper embryonic care is taken and protection is given, and greater survival chances of young ones in viviparous animals. Then the zygote in flowering plants. The zygote in flowering plants is formed inside the ovule of flowering plants, where after the fertilization, sepals, petals, and stamens of the flower will wither and fall off. Zygote develops into embryo, ovules into seed, and ovary into fruit. Then the thick wall of the fruit is called as pericarp. It's called as pericarp. So uh, we'll stop here, students. We'll solve a few MCQs. Just a second. One second. Um, Sir, won't monoecious plants be considered under sexual reproduction according to uniparental condition? See, it is not unisexual reproduction. What do you mean by unisexual reproduction there? So, monoecious plants are bisexual. Understand this. Monoecious plants are those plants which have both male and female structures with them. So, remember that. So, monoecious plants have both male and female structures with them. Okay. So, what is there? Nothing like unisexual reproduction. Okay. So, here monoecious plants have both male and female structures. So, they are bisexual. Bisexual. Okay. Then, sir, what is oviparous? I have told you about it. We'll solve questions now. Which of the following groups is formed by hermaphrodite organisms? What do you mean by hermaphrodites here? Here, hermaphrodites are those organisms which have both male and female structures with them. Okay, which have both male and female structures with them. So here, earthworm is a hermaphrodite. Yes, tapeworm is a hermaphrodite, but housefly is not a hermaphrodite. Frog is not a hermaphrodite. Okay, because they are unisexual. You have male and female frogs. So earthworm fine, tapeworm fine. Housefly, seahorse, no. Seahorse is basically a Piscean. It can't happen. So earthworm is correct. Leech is correct. Roundworm, no. The last one, earthworm, leech, tapeworm, sponge. Yes, D is the correct answer. D is the correct answer. Very good. D is the correct answer. We'll go to the next question. Which of the following has the longest lifespan? Think and answer. Which of the following has the longest lifespan? Okay. So we have different answers here. See, let's go. Let's start one by one. Okay, let's start one by one. What about elephant? We'll go from here. Elephant is somewhere around 60 to 90 years. Tortoise, sorry, parrot. Parrot can be 140 years. Tortoise. Tortoise is one of the highest. How much is it? It is 100 to 150 years. Banyan tree. See, banyan tree is close to 200 years. 200 years. So A is the correct answer. A is the correct answer. Why? Because there is a banyan tree, it keeps on spreading. Okay. So we'll go to the next one. Asexual reproduction. Asexual reproduction is seen in members of kingdom. Think and answer this question. Think and answer. See, monerans, they're not asking absolute asexual reproduction. Think, monerans have asexual reproduction. Even plants, some of them have asexual reproduction. Okay. Animalia, some of them have asexual reproduction. Example, in the case of sponges. Okay. They have asexual reproduction. By fragmentation, they grow. So D, all of the above. Now students will come up, sir, we can't say because in Monera you sexual also. They have just asked. Asexual reproduction is seen in members of the kingdom. All of the above is the correct answer. Then, please answer the fourth question. Sensor. Okay. Okay. We'll just see. Read the statement and pick the correct ones. So good, many of you have answered properly. Conidia are asexual propagules restricted to kingdom fungi. Okay, fine. Then a piece of potato tuber having at least one eye or node is capable of giving rise to a new plant. That is also correct. Ginger propagates vegetatively with the help of underground roots. Wrong, it is a stem modification. It can't be with underground roots. Fleshy buds which take part in vegetative propagation are called bulbils present in Dioscorea and agave. That is correct. So C is the correct answer. So C is the correct answer. Very good. Very good. Good. Many of you have answered. Good. Then the growth phase of an organism before attaining sexual maturity is referred as. Please answer. Okay. Okay. Yes. Juvenile phase is also correct. If we take animals into consideration, it is called juvenile phase. Plants, it is vegetative phase. It is both A and B. Good. Then we go to the next one. Please answer. Students, read the following statement. Many plants are propagated vegetatively even though they bear seeds. Sweet potatoes multiply vegetatively by root tubers. Please answer. Okay. See, many plants are propagated vegetatively even though they bear seeds. That is true. Okay. Yes, many plants they can be propagated vegetatively even though they bear seeds. Why? Because if you take onion bear seeds, it does produce seeds. At the same time, it can be propagated vegetatively also. So A is the correct answer. Sweet potato multiplies vegetatively by root tubers. Sweet potato, see, understand this, it is not potato, it is sweet potato. Sweet potato is a root modification and not a stem modification. So remember this. So think and answer. Which is the correct answer? Yes, B is the correct answer. B is the correct answer. B is the correct answer. Then we go to the next one. Geme are means of vegetative propagation in. Okay. Answer, students. So you've heard about Geme in the last year. So in plant kingdom, Gemma cups. So no, it is not colocasia. Here, students will get confused with the gemmules. It is Geme. It is Markantia. It is Markantia. In Markantia, you can see Gemma cups. Okay. So seventh one, D is the correct answer. It is not Spirogyra, it is not Marsilia, it is not Colocasia. It is Markantia. So usually students, when they get this name from this chapter, they usually relate it to gemmules. Gemmules are found in sponges. Then stem cuttings are often treated with Na. Na is a auxin. Na is a auxin. Are often treated with Na before sowing in order to promote. Think and answer, students. So when you put it into soil, what should happen first? So that they'll be able to absorb water. Answer. Okay. So yes, very good. It is for rooting. So if they're dipped in oxen and then placed in soil, they'll immediately start rooting because it will promote cell division. So remember this. Once rooting take place, they'll be able to grow. We'll go to the next one. Leaf and stem cuttings are sown. Answer. Think and answer, students. Very good. See, uh, remember this, students. There's a technique. So every time we take a stem cutting from a plant, it has to be sown in soil. Let us imagine there is a plant like this. Okay, with okay. So these are roots and all that. So we take a part and we have to place it somewhere, and the apical end should always face upwards. So remember this, the apical end should face upwards. Okay, so vertically, so it can't be laterally at all. Okay, it can't be laterally. Laterally means placing like this. We don't place a stem cutting like this. Okay, so vertically with morphological apical end upwards, that is correct. Second one we'll just see vertically with morphological basal end upwards. This is wrong. Apical end should be upwards. So A is the correct answer. A is the correct answer. Then 10th one, please answer. Clone is. Please answer. Clone is. Yes. They can be descendants of the same parent. Yeah. If it is born by asexual reproduction. Okay.
So even though if it is descendant of a single parent, and if it is bisexual reproduction, if it is bisexual reproduction, so if it is bisexual reproduction, then it can't be a clone. So remember this, what are clones? Clones are genetically and morphologically, they should be similar to the parent. Okay, they should be similar to the parent. But what is happening here, they're telling descendants of a single parent. Just because it is descendant of a single parent, we can't say it is a clone. Okay, so if even though if it is from a single parent, they would have undergone sexual reproduction, so the chances are less that it could be a clone. Fusion product of two parents, this is absolutely wrong. This is absolutely wrong. Formed from parthenogenesis. So listen here, if it is formed from parthenogenesis, it is formed out of a single gamete, that is from the mother, that is from the mother. Okay, but there is a formation of gamete, so it should not be formed from a gamete. So D, understand this, none of the above is the correct answer. They should be born from a single parent, but by what is the prerequisites? It should be asexual reproduction. It should be by asexual reproduction. So remember this, this is very, very important. Then sexual reproduction involves two parents, each of which contributes. So what does each of one contributes? Please answer. Very good. Each of will contribute a gamete. Each of which will contribute a gamete. Then basic forms of asexual reproduction are fission, budding, gemulation, and fragmentation. Binary fission is common among. Answer. Very good. It is in bacteria and protozoa. Then binary fission is seen in. Please answer. Binary fission is seen in. So where is binary fission seen? Answer. See, Linearia it is not possible. Think and answer.
So here paramecium is the correct answer. Paramecium is the correct answer. Go to the next one. Chrysanthemum multiplies vegetatively by. Answer. Chrysanthemum multiplies vegetatively by. So chrysanthemum multiplies vegetatively by sucker. Then internal buds of freshwater sponges are otherwise called. Please answer. Very good. They are by gemmules. So in sponges it is gemmules. So students will stop here. We'll start with microbes in human welfare. Okay, just give me a second. One second. So we'll study microbes in human welfare. In microbes in human welfare, we'll be studying about microbes in household products, microbes in industrial products, microbes in sewage treatment, microbes in production of biogas, biocontrol agent, and as biofertilizers. We'll go quickly through each concept. Microbes in household products. Here you have to know
the name of individual microbe and its composition onto what it produces. That is very important. The first one is lactobacillus, okay, also called as lactic acid bacteria. Lactic acid bacteria. So remember this. So what do? They convert milk to curd. So why is it important? A small amount of curd plus fresh milk will convert it into curd and increases the vitamin B12. This is very important in this part. Please remember this vitamin B12. It helps to check pathogens. Then, so why is it used? The next one, dosa idli products. Please remember this. So puffing off all this. Then baker's yeast, Saccharomyces cerevisiae. It is used to make bread by fermenting dough. So baker's yeast, remember this. Then toddy is made by fermenting sap from plants. Microbes are used to ferment fish, soya bean, and bamboo shoots to make foods. Remember this. This is also very important. Then production of cheese. Example, in Swiss cheese, we have lot of holes in it, which are by production of CO2 by Propionibacterium sharmani, which is a bacterium. When Roquefort cheese is ripened by growing specific fungus, which gives a particular flavor. Then microbes in industrial products. So here in industrial products, all that we are looking at is beverage and antibiotics at the industrial scale. Fermented beverages. Saccharomyces cerevisiae, also called as brewer's yeast, which is used to ferment malted cereals and fruit juices to produce ethanol. So there are two types of beverages here: wine and beer, which are produced without distillation; whiskey, brandy, and rum, which are produced by distillation of fermented broth. Distillation of fermented broth. Then antibiotics. Antibiotics are chemicals which are produced by some microbes and can kill or retard the growth of other disease causing microbes. Other disease causing microbes. They are used to treat plague, whooping cough, diphtheria, leprosy, etc. Then penicillin. It is the first antibiotic discovered by Alexander Fleming. Please remember this. It is produced by a mold, Penicillium notatum, which was growing on unmarked culture plates around Staphylococcus, which could not grow. So he extracted penicillin from it. Okay. So Ernest Chain and Howard Florey established its full potential as an effective antibiotic. Okay. So they got Nobel Prize. Then please do remember organic acids, which are very important. Aspergillus niger helps in production of citric acid, Acetobacter aceti with acetic acid, Clostridium botulicum butyric acid, Lactobacillus lactic acid. So chemicals and other bioactive molecules. Alcohol, Saccharomyces cerevisiae, is used to produce ethanol. Then enzymes which are produced, lipases which are used in detergent to remove oil stains from laundry, pectinases and proteases to clarify bottled juices, streptokinase produced by streptococcus used as clot buster to remove clots from the blood vessels of patients who have myocardial infarction. Then cyclosporin, cyclosporin A, which produces a immunosuppressive agent which is used in organ transplantation. So it is produced by Trichoderma polysporum, which is a fungus. Then statins produced by Monascus purpureus, which is a yeast. It is used as a blood cholesterol lowering agent. It is used as a blood cholesterol lowering agent. It inhibits the enzyme responsible for synthesis of cholesterol. It inhibits the enzyme responsible for synthesis of cholesterol. Then use of microbes in sewage treatment. So we'll look at sewage treatment. Sewage is basically municipal waste that contains large amounts of organic matter and microbes. Sewage is treated in sewage treatment plants. Sewage is treated in sewage treatment plants to make it less polluting, which includes two stages. One is primary treatment, the other one is secondary treatment. This, in the primary treatment, it is the physical removal of particles. It includes removal of floating debris by sequential filtration, then removal of grit, soil, and small pebbles by sedimentation. So remember this, filtration and sedimentation. Then all solids that settle form the primary sludge, and the supernatant forms the primary effluent. Primary sludge and primary effluent. Then the primary effluent is then pushed on to aeration tanks where it is constantly agitated, which induces the growth of aerobic microbes into flux. Then they consume organic matter in the effluents, reduce the BOD of the effluent. This is very important. What is BOD? BOD is the amount of oxygen consumed by bacteria to oxidize organic matter. Remember this, it will only oxidize organic matter in 1 liter of water. BOD is a measure of organic matter present in water. The greater the BOD, more is the polluting potential. Then after that, it is then pushed on to, then the effluent is pushed on to a settling tank where sedimentation of flux takes place. Okay, sedimentation of flux takes place to form activated sludge. The activated sludge is pumped back into aeration tank to serve as inoculum. Remember this, so that they can be used again in the previous tanks. Then anaerobic sludge digesters are used where they digest the bacteria and the fungi in the sludge. This produces gases like methane, H2S, and carbon dioxide. So which would be combined to form the biogas. Then we go to the next concept. Ministry of Environment and Forest has initiated Ganga Action Plan and Yamuna Action Plan to save water from water pollution. And we go to microbes in production of biogas. So a mixture of gases, mainly methane, produced by microbes such as methanogens. Methanogens grow anaerobically on cellulosic material to produce methane along with carbon dioxide and hydrogen. Remember this, methanobacterium, which is found in the anaerobic sludge and rumen of cattle. What do you mean by rumen of cattle? So cattle have 4-chambered stomach: rumen, reticulum, omasum, and obomasum. Okay, rumen of cattle, which help in cellulose digestion. So the cattle dung or the gobar contains these bacteria. So the dung is used for generation of biogas. So there's some amount of cellulose still left in the dung, which will be converted to biogas. So look at this. What does it consist? A biogas plant consists of a concrete tank which is 10 to 15 feet deep to collect bio-wastes and slurry of dung. A floating cover, can you see here? A floating cover is placed over the slurry, which keeps on rising as the gas is produced in the tank. So once it is completely risen, what does it mean? That the tank is completely filled with biogas. Okay, then after this, there is an outlet for the supply of biogas. Okay, and there's an outlet to remove spent slurry, which can be used as fertilizer. And what are the uses of biogas? Biogas is used in cooking and lighting. So it was developed by Indian Agriculture Research Institute and Khadi and Village Industries Commission. So they developed this technology of biogas in India. We go to the next one, microbes as biocontrol agents. So
It is the use of biological methods for controlling pests. Chemical pesticides and insecticides are harmful to all organisms and they cause pollution. The chemical pesticides kills both useful and harmful life forms. This is very important. So what are the examples of biocontrol agents? Ladybird beetle to get rid of aphids, dragonflies to get rid of mosquitoes. Okay. Then the use of Bacillus thuringiensis to control butterfly caterpillar. Remember this, to control butterfly caterpillar. To control butterfly caterpillar. Then it is available in sachets as dried spores, which are mixed with water and sprayed onto vulnerable plants. So this is a very important one. And also remember this, scientists have introduced Bacillus thuringiensis toxin to genes into plants. Example, BT cotton. Example, BT cotton. We go to the next one. Trichoderma polysporum is, so here they are found in root ecosystem. They control several plant pathogens. They control several plant pathogens, especially, look at this, with baculoviruses or nuclear polyhedrovirus. They attack insects and other arthropods. They are suitable for species-specific narrow spectrum insecticidal activity. So they'll kill only those insects which are harmful to us. They'll not kill the harmless insects. Okay. This is desirable when beneficial insects are being conserved in to aid integrated pest management, program, or when an ecologically sensitive area is being treated. This is very important. Okay. Then we go to microbes as biofertilizers. So biofertilizers are organisms that enrich the nutrient quality of the soil. Example, bacteria, fungi, cyanobacteria, etc. Then next one, rhizobium. It's a bacterium, is seen in the root nodules of leguminous plants. Understand this, root nodules of leguminous plants, they fix atmospheric nitrogen. Okay. So rhizobium, how are they? They are found inside the root nodules of leguminous plants. Whereas there are free living organisms also in the soil, azospirillum and azotobacter. So they enrich the nitrogen content of the soil. They enrich the nitrogen content of the soil. Then the next one, mycorrhiza. So it is a symbiotic association of some fungi. Example, glomus with plants. Understand this, symbiotic association of some fungi. Example, glomus with plants. So what do they help in? The fungus gets food and the plant will get nutrition. Okay, from the fungus to some extent, very small extent. I'll tell you what is that. They absorb phosphorus from soil and passes it onto the plant. So helps in absorption of phosphorus. Gives resistance to root-borne pathogens. So gives resistance to root-borne pathogens and tolerance to salinity and drought. They help in plant growth and development. All three very, very important. Then cyanobacteria, that is blue-green algae. They're basically autotrophic microbes. They can fix atmospheric nitrogen. Example, Anabena, nostoc, and oscillatoria. Okay. In paddy fields, cyanobacteria serve as biofertilizers. It acts organic matter to soil and also increases soil fertility. Understand this. Cyanobacteria, blue-green algae, they are prokaryotes. Understand this. Anabena, nostoc, and oscillatoria, all three. So this brings you to the end of the synopsis of this chapter. We'll solve MCQs now. Just a second, students.
Answer, students. Big holes in Swiss cheese are made up by a okay. So bacteria producing large amount of carbon dioxide. Very good. Bacteria producing large amount of carbon dioxide. Then technology of biogas production from cow dung was developed in India largely due to efforts of okay. See, it is not gas, it is not oil and natural gas. It is Indian Agriculture Research Institute and Khadi and Village Industries Commission. Very good. Then mycorrhiza does not help the host plant in. Mycorrhiza does not help the host plant in. Answer. So here they have not asked about glomus. So specifically about glomus. So think and answer mycorrhiza. So what do they help in? Okay. So the root and taproot in some gymnosperms are associated with fungus. They are called as mycorrhiza. Example in the case of pinus. Okay. And some of the others are associated with cyanobacteria, that is blue-green algae. Those are coralloid roots which are in cycas. So think and answer. See, phosphorus uptake, correct. Tolerance to drought, root pathogens, correct. Does not help, they've asked. See, does not help. So it helps in phosphorus uptake, it helps in tolerance to drought, it helps in resistance to root pathogens. That is true. It does not help in increasing the resistance to insect. So D is the correct answer. They've asked does not. Does not. Okay. So it enhances phosphorus uptake, it increases tolerance to drought, it enhances resistance to root pathogens. The one it does not do is resistance to insect. Does not. D is the correct answer. Then BOD of wastewater is estimated by measuring the amount of answer, students. BOD of water is estimated by measuring the amount of think and answer. See, total organic matter, yeah, but all that organic matter need not be degraded by microbes. No. Okay. It is biodegradable organic matter. So what do we say? Biological oxygen demand is the amount of oxygen required to degrade organic matter present in water. So look at it. How can it be oxygen consumption? So if there is a fish in it, based on the oxygen consumption, can we say it is BOD? Wrong. Oxygen evolution is wrong. It's not with plants. So B is the correct answer. Think biodegradable organic matter. There should be organic matter that should be biodegraded by microbes. So this is a tricky question again. Okay. See, the question looks very simple. As soon as we look at it, the only thing that comes to us is this and this. Oxygen evolution will be sure that it is wrong. but B is the correct answer. Okay. The residue left after methane production from cattle dung is please answer. Yes, very good. They're used as manure. They're used as manure. Then Streptomyces venezuelae yields. Streptomyces venezuelae yields. Answer. Okay. Okay. 1 second, students. See, it is actually chloramophenicol. It is actually chloramophenicol. Okay. Remember this, chloramophenicol. So we'll go to the next one. Free fungus. Trichoderma can be used for. Free fungus, free living fungus. Trichoderma can be used for. What can trichoderma be used for? Huh? Think and answer. See, it is not disease, okay? Biological control. It is not biological control of plant disease. See, if there is a insect feeding on it, it is not a disease. They are being pathogenic. Antalla, they are parasitic. So think, it is B is not the answer. Trichoderma is a free living fungus used to control several plant pathogens. Control of plant pathogens. Which are they? Controlling butterfly, caterpillars, sure. See, BT is used for controlling, it is for killing insects, for killing insects, for killing insects. Then methanogens are found in. Please answer. Methanogens are found in. Okay. Okay. All of these. Very good. It is found in all of these. Then Monascus purpureus is yeast commercially used for the production of Monascus purpureus. Please answer. So statins are produced by Monascus purpureus, which are blood cholesterol lowering agents. Very good. They are blood cholesterol lowering agents. Good. Then match the following. Okay. Okay. Citric acid. Citric acid is produced by Aspergillus niger. So citric acid is produced by Aspergillus niger. Okay. The next one, cyclosporin A. Cyclosporin A. So cyclosporin A, it is basically used as a immunosuppressive agent. So cyclosporin is got from Trichoderma polysporum B. Okay. The next one, statins. So statins are very important in lowering the blood cholesterol level. They are produced by Monascus purpureus. C. Gobar gas is by Methanobacterium. So D. So A is the correct answer. A is the correct answer. Done. So we go to the next question. Roquefort cheese is ripened by using a. So what do we use in producing cheese? Just a second, students. Just think and answer. So answer, students. So Roquefort cheese, what is it? Please do answer. So it is a specific type of fungus. It's a specific type of fungus. Very good. We'll go to the next one. Azula enriches rice fields with nitrogen due to its association with. So Azolla will enrich rice fields due to its association with. Answer, students. So very good. It's association with nostoc. Nostoc. What is false about antibiotics? What is false about antibiotics? Please answer. Just give me a second, students. It antibiotics, the term was coined by Waltzman. That is absolutely right. They are produced by microorganisms. That is also right. Produce allergy in some person. Yes, it is true, because penicillin produces allergy in many individuals. It is not good for many individuals. So cure all the diseases is the right answer, because antibiotics can't cure all the diseases. We'll go to the next one. Yeast is employed for the production of. Yeast is employed for the production of. So curd is wrong. We need lactobacillus. Cheese, certain fungi and bacteria. Acetobacter aceti for acetic acid. Ethyl alcohol is correct. So yeast is used, that is Saccharomyces cerevisiae is used for production of ethyl alcohol from plant sap. Plant sap. And malt, that is from germinating seeds. that is from germinating seeds. Then symbiotic association between fungus and seed plant. Please answer, students. Symbiotic association between fungus and seed plant. No, commensalism is wrong. What happens in commensalism? One of the individual is benefited and the other one is neither harmed nor benefited. So helotism is wrong. It is endomycorrhizae. Endomycorrhizae is correct because they enter into the tissue. Endomycorrhizae. Identify the fungus with medical importance. Identify the fungus with medical importance. Please answer. Yes, it is Penicillium notatum, which helps in production of antibodies. Then probiotics are, what are probiotics? So probiotics is something we daily take it in our diet, almost every one of us. Very good. Live curd has lactobacillus in it. Curd. So we have very good yogurt by increasing. Very good. It is vitamin B12. Vitamin B12. Good. Then cyclosporin A is used to. What is cyclosporin A used as? Okay. It does not regulate blood pressure. It does not remove blood clots. Blood clots are removed by clot buster, that is streptokinase. They do not treat respiratory disorders also. They basically act as an immunosuppressive agent. So they inhibit the rejection of transplanted organ. They act as immunosuppressive agents. So remember this, they act as immunosuppressive agents. Then citric acid is obtained from. Citric acid is obtained from. Good. Citric acid is obtained from Aspergillus niger. Aspergillus niger. Good. Then statins used for lowering blood cholesterol level are extracted from. So statins are obtained from Monascus purpureus. Monascus purpureus. Answer, students. So it is basically it is not an algae. It is not an algae. Think it is not bacteria. It is not virus. It is a yeast. It is a yeast. It belongs to a category of mold. Aerobic breakdown of organic material is known as. Aerobic breakdown of organic material is known as. They've asked aerobic. Think and answer, students. No. Fermentation is absolutely wrong because that is anaerobic breakdown. Decomposition and decay, both are right. So the correct answer is D. Both A and B. Both A and B. Fermentation is anaerobic. Fermentation is absolutely anaerobic because the end products are lactic acid and or ethyl alcohol. What happens when we inoculated rhizobium in wheat field? So they've told wheat field, it is not a rice field. Understand this. So both here fertility of soil, let me make this increases. Sorry, there's a mistake, increase. Think and answer. See, obviously fertility of the soil will not decrease 200%, it won't decrease. Why? Rhizobium is there. Think and answer. See, rhizobium usually associates itself with legumes. Think that in, keep that in mind. Think and then answer. So these are questions which will try to confuse you. So they here will study about rice field. Here they have given wheat field. So very good. There is no increase in production. There is no increase in production. See, the fertility of the soil, yes, does increase to a certain level, but not much of difference is seen. There's a lot of increase in production, no increase in production. Very good. Then, which one of the following pairs are correctly matched? See, they've asked correct ones. See, remember this, correctly match. Rhizobium, parasites in roots of leguminous plants is wrong. A is wrong. Why? Because they are not parasites. They are symbiotic. They are symbiotic. Both of them are benefited here. Mycorrhizae, yes, mineral uptake is true because they help in uptake of phosphorus. Yeast, production of biogas, it's wrong. Myxomycetes, disease ringworm, wrong. B is the correct answer. Mycorrhizae, mineral uptake from soil. Mycorrhizae are symbiotic association between fungus and roots of higher plants. Bacillus thuringiensis strains have been used for designing novel is new. Yes, it is bioinsecticidal plants. Very good. Bioinsecticidal plants. A common biocontrol agent for control of plant disease is think we had solved a very similar question before. So see, Bacillus thuringiensis, no, it's wrong. Very good. Trichoderma is correct. Trichoderma will control several plant pathogens. So baculovirus, wrong. So what do baculovirus do? They attack insects and arthropods. So it's wrong. Bacillus thuringiensis will be on all this butterflies and moths. Okay, the larvae of them. So butterfly caterpillars and all that. Glomus, there's a symbiotic association. So trichoderma is the correct one. Then a common enzyme produced commercially from Saccharomyces cerevisiae is a common enzyme produced commercially from yeast is it is not lactase. Think Saccharomyces cerevisiae. It is invertase. It is invertase. Organic farming does not include. Organic farming does not include. What doesn't it include? Very good. It doesn't include chemical fertilizer. All that we use is green manure, farmyard manure, compost, and waste indigenously produced in the same farm. Farmers have reported over 50% higher yields of rice by using which of the following biofertilizers? See, it can't be Bacillus thuringiensis. Why it is insecticidal? Legume rhizobium. So here it's all rice. So B is wrong. Mycorrhiza is wrong because it is symbiotic association of fungus. It is cyanobacteria. Cyanobacteria. So we have done this in the synopsis. Cyanobacteria. Then the next question. Which of the following statements regarding baculovirus as biocontrol agents is or are correct? So a majority of baculovirus used used in used as biocontrol agents are included in the genus nucleopolyhedrovirus. Yes, this is absolutely right. They are from nucleopolyhedrovirus. Then infection with baculovirus occurs when susceptible hosts, example, some specific insects, eat the virus present on the foliage and become dead. That is also true, absolutely true. Okay. Then these are important in organic farming because of the specific action on harmful insects without causing any damage to beneficial insects as well as to the environment. Even that is absolutely true. The correct answer is all of the above. All of the above. Okay. Students will stop here. In the next class, I'll be taking up living world and biological classification. I'll be taking up living world and biological classification. Sir, I'll be ending the session now.
Sure, sir. I will end the session now. Yeah.
Hello, sir.
I mean,
I'm
sir.
not able to. Sir.
How are you, sir?
I'm fine, sir. Absolutely fine.
Okay, sir. Fine, sir. I will end the session.
Yeah,
Sir,
sure.
after this class, I just want to talk to you for a minute.
Yeah, sure, sir. Sure.
Okay. Thanks, sir.
Thanks.
Bye. Bye, sir. ಈಗ ಮೀಟಿಂಗ್ ಎಂಡ್ ಮಾಡೋದಾ ಇಟ್ಕೊಳ್ಳೋದಾ? ಇದ್ದಾರೆ ಹೋದ್ಮೇಲೆ ರೂಮ್ ಲಾಕ್ ಆಗುತ್ತೆ. ಪಾರ್ಟಿಸಿಪೆಂಟ್ಸ್ ಹೋದ್ರೆ ಇದೇನೋ ಅಲ್ಲಿ ರೂಮ್ ಲಾಕ್ ಇದೆಯಾ? ಸರಿ 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 ಸರಿ. ಹಲೋ ಸರ್. ಸರ್ ಈಗ ನೆಕ್ಸ್ಟ್ ನಿಮ್ಗೊಂದು ಕ್ಲಾಸ್ ಇದೆ ಅಲ್ವಾ? ಇದು ಅದು ಬ್ಯಾಚ್ 2. ನಾಟ್ ಮೆನಿ ಸ್ಟೂಡೆಂಟ್ಸ್ ಆರ್ ದೇರ್ ಇನ್ ದಟ್ isn't it? ಇವತ್ತು ಪ್ರಜ್ವಲ್ ಬರಲ್ಲ. ಕ್ಯಾನ್ ಯು ಮ್ಯಾನೇಜ್ ವಿತೌಟ್ ಹಿಮ್? ಹಿ ಈಸ್ ಇನ್ GT ರೈಟ್ ನೌ. ಅದಕ್ಕೆ ಓಕೆ 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 ಫೈನ್ ಸರ್ ಫೈನ್ ಸರ್. ಹಾಗೆ ಸರ್. ಹೌದು.
We can start, sir.
You can start, sir.
Thank you. Welcome back, students. Good afternoon to one and all. So we will continue with where we had left yesterday, students. I believe we were discussing moving charges and magnetism in the previous class. Let us continue with some more concepts related to magnetism and matter, and after that we will quickly look into a few questions that are there from the chapter. Post that, students, I will be discussing today. Post that I will be discussing ray optics. All right. Let us begin our session. The students, in the previous class, we had seen about how we can interplay between electric field and magnetic field. Also, we had seen about breaking of bar magnet, where if you break a bar magnet along the vertical, then magnetic moment becomes m by 2, whereas moment of inertia becomes I by 8, and therefore we saw time period will become half of the original time period for the entire bar magnet. Likewise, we saw that if you cut the bar magnet along the horizontal, magnetic moment changes as m by 2. Along with that, moment of inertia also changes as I by 2. So from there, students, we got T dash to be equal to T. I think this is where we had stopped. We had also defined Earth's magnetic field. We had written down a few relations for finding the horizontal component, the dip, and the declination. Students, with that, let me next discuss some terms and terminologies. First one of that sort is called magnetization, or also called as intensity of magnetization, which is magnetic dipole moment per unit volume. Then we have magnetizing field. Magnetizing field. Students, magnetizing field is denoted by the letter H. All right. This is denoted by the letter M. Magnetizing field is denoted by the letter H, which is defined as the magnetic field divided by the permeability of the material. Or in other words, students, if B naught is the magnetic field in air, then it will become B naught divided by mu naught. All right. So these are some associated terms and relations required in moving charges and magnetism and matter. Next, students, we have permeability, where permeability is defined as or written as mu, which is product of mu naught into mu r for a given medium. Lastly, we have susceptibility, where susceptibility is denoted by the letter chi. Susceptibility is nothing but magnetization per unit magnetizing field. These are some relations, students, where if you substitute your magnetic field due to dipoles, magnetic field due to dipoles as Bm, then it is mu naught into M. Magnetic field due to the solenoid, we write it as mu naught into H. Therefore, net magnetic field turns out to be B naught plus Bm when a material is placed inside a solenoid. Students, on simplifying this, we get a relation mu r is equal to 1 plus chi. So these are the required formulas for solving MCQ, students. These are all the required formulas for solving MCQs connected to magnetism and matter. Having said that, let me quickly give you some key highlights between your ferromagnets, paramagnets, and diamagnets. So if I take ferromagnet, students, they are strongly attracted to magnetic field. they are very strongly attracted. If you take paramagnets, they are attracted but not very strongly. They are weakly attracted. Whereas diamagnet, when you see, they are weakly repelled by the magnetic field. All right. Therefore, ferromagnets move from a region of strong magnetic field to, weak magnetic field to strong magnetic field. If you place a ferromagnet, they move from weak magnetic field to strong magnetic field. Whereas paramagnets also is same. They also move from weak magnetic field to a region of strong magnetic field. Whereas when it comes to diamagnets, it's ulta. It moves from a region because it repels, na? It moves from a region of strong magnetic field to weak magnetic field. So students, we say that the reason for ferromagnets are domains which are a collection of dipoles having same moment. Dipoles with same moment. When they all align in a direction, they contribute to magnetism. Likewise, we have paramagnets also. The reason for paramagnets is said to be unpaired electrons. Unpaired electrons are the reason for paramagnets. And diamagnets are the ones which repel. And that is said to be having no unpaired electrons. Diamagnets are said to have no unpaired electrons. Coming to the permeability, students, we say relative permeability is very, very, very large for ferromagnets, roughly of the order of 1,000. Whereas for paramagnets, it is greater than 1, but not as big as ferromagnets. For diamagnet students, permeability is positive, but less than 1. Permeability is positive, but less than 1. All right. Now coming to your susceptibility, students. Susceptibility is also very, very large. So we say susceptibility is large and positive. Here the susceptibility is small, but still it is positive. Students, in the third case, susceptibility is much less than 1. In fact, it's negative. So we write it as less than 0. It is negative. So here, students, it is small and negative. So these are some relations that are between so this is for your diamagnet students. Lastly, we next look into our one of you are asking if they have tangent galvanometer. In fact, we have not seen any question of late, so it is not there in the syllabus bulletin. We have not seen any questions of late. Okay. So students, lastly, we have Curie's law, where Curie's law says that the susceptibility of a material is inversely proportional to absolute temperature for ferromagnets and paramagnets, but it does not obey for diamagnets. This Curie's law does not obey for diamagnets. So students, if the temperature increases, susceptibility will decrease. Therefore, for paramagnets, we say paramagnets, they keep losing their magnetism with rise in temperature. They keep losing their magnetism. Ferromagnets, on the other hand, students, they are slightly different. They also keep losing, no doubt. But at one point, called as critical temperature, the ferromagnets will become diamagnets. The ferromagnets will become diamagnets at a point called as critical temperature. So therefore, For your paramagnets, the relation for Curie's law is that chi is equal to some constant K divided by absolute temperature. But for ferromagnet students, that chi is not just equal to, that chi is not equal to just K, it is K divided by T minus Tc, where Tc is called as Curie's temperature, where Tc is called as Curie's temperature. Ferromagnets will not turn into para, they turn into dia. Ferromagnets turn into diamagnets. All right. And that temperature at which it happens is called as critical temperature. Okay. So these are some ideas connected to Curie's law, students. Lastly, we will be discussing hysteresis law, where if you magnetize a material, then the magnetic field induced in it is given by this curve, where after a point it attains saturation. If you reduce the magnetization, there comes a point where it will retain a magnetic field. Students, this is what we call as retentivity, where retentivity is what? It is a property of a material to retain magnetic field. Further, if you reduce, it demagnetizes. The amount of reverse magnetic field required to demagnetize is called coercivity. Amount of reverse magnetic field required to demagnetize is called coercivity. On further increasing, it will magnetize in opposite direction and then get demagnetized, and the process keeps on repeating. Students, this is called as hysteresis curve. This is called as hysteresis curve, or also called as BH curve. One more name, students, it is called as curve for magnetization and demagnetization. Magnetization and demagnetization. So one of you are asking me to define what is coercivity. I will repeat. Coercivity is the amount of reverse magnetic field. Coercivity is the amount of reverse magnetic field required to completely demagnetize a material. All right. Now, what is the significance of this BH curve? Students, if retentivity, all right, and coercivity is high of a material, if retentivity and coercivity is high, we use it for making permanent magnets. All right. Whereas if retentivity and coercivity is low, then we use it for making electromagnets. These are the two applications of retentivity and coercivity. Okay. Now, we also see that the area under the graph, this area under the graph also has a significance. Students, this area under the graph of a BH curve, this area under the graph of a BH curve, it signifies hysteresis loss. Meaning, it will tell me hysteresis loss, meaning it is the heat lost or energy lost during a cycle of magnetization and demagnetization. Heat lost or energy lost during the cycle of magnetization and demagnetization. What does it mean, students? See, I'll give you an example. If I am supposed to manufacture a material that is not supposed to get heated up, I want to manufacture a material that is not supposed to get heated up. Students, I perform an experiment and I get two graphs. One looks like this. And for another material, it looks like this. Now, can one of you tell me which graph will you use? Will you use graph A or graph B material for having minimum loss? Which one will you use to have minimum loss? Wonderful. So the one which has least area, we use it for having minimum heat loss. And therefore, the type of material is called as soft iron core, which is used in transformers. Soft iron core, which is used in transformers. So this is some application, students. All right. These are some applications related to our ferromagnet and paramagnet. Shivank, it is not a B, it is A because area is minimum. Okay, fine. Now, having dealt with this, let us quickly look into some questions connected to this. Let's start with the first one. First question. The ratio of magnetic field due to a small bar magnet at the end-on position, this is the end-on position, to the broad position. This is the broad position. Is the question. Students, this is axial point, right? This is equatorial point. We know for a fact that axial point is given by mu naught by 4 pi, right? 2m by r cube. Whereas equatorial point, what is the relation, students? It is mu naught by 4 pi minus m by r cube. So if I take ratio between these two, I'll get 2:1. Am I right? Therefore, right answer is option D. Did all of us understand? Next question, students. The potential energy of a bar magnet, which has magnetic moment M, is placed in magnetic field B such that it makes an angle theta in the direction of magnetic field. Which is the right option, students? Which is the right option? That is correct. It is minus mb cos theta. This is the definition. It will be minus mb cos theta. Next question, students. The time period of oscillation for a magnetometer is 2 seconds. The period of oscillation of the magnet, if moment of inertia becomes 4 times that of the first magnet. Students, time period is directly proportional to root of I. Therefore, can I say T by T dash will be equal to root of I by root of 4I. On simplifying, we will get 1 by root 4, which is 1 by 2. Quite clearly, T by T dash is 1 by 2. What is the answer then, students? What is the answer then, students? So from here, T dash, if you cross multiply, T dash will become 2T, where T is given as 2 seconds. Therefore, T dash becomes 4 seconds. Is that clear to all of you, students? It's not option D. Some of you have told option D. Please check the calculation. It is option. Very good. Very good, students. Next question. What is the right graph between for paramagnetic material? Students, we all know chi is inversely proportional to temperature. Chi is inversely proportional to temperature. So students, if chi versus T was plot, then inverse graph would have been like this. This is chi versus T. But they have not plot chi versus T, they have plot chi versus 1 by T. That is why, students, option B is not correct. chi versus 1 by t, meaning if chi is plotted on y-axis, 1 by t is plotted on x-axis, then it represents the form y is equal to mx plus c. Then this equation represents the form y is equal to mx plus c, which is a straight line passing through the origin. Straight line passing through the origin. So which is a straight line passing through origin, students? Option A. Straight line passing through origin is option A. Are we all clear with this idea? Did we all understand, students? Good. Next question. Domain formation is a necessary feature for theory question. Lot of theory questions from this chapter, students. That's why I have pulled up them. Very good. Domain theory is necessary for ferromagnets, not for all of the above. Ferromagnetism. Next question. The magnetizing field of a ferromagnet is increased. What happens to its permeability? Students, magnetizing field meaning H. All right. Permeability meaning mu r. Students, permeability is nature of the material. The nature does not depend on external factors. Nature does not depend on external factors. Therefore, students, we can say from here, option C is true. It will not depend on permeability. Remains constant. Are we all clear? It will not depend, therefore it remains constant. So I think with that, we will end up the questions on more magnetism and matter, students. We have covered good number of questions, especially to do with the theory. Now I am going to start with ray optics. It's a very lengthy chapter, students. When it comes to MCQs, there are a lot of problem types that are discussed in ray optics. That is why, students, today I am going to strictly restrict myself to reflection. I'm going to strictly restrict myself to reflection. All right. In reflection chapter, I am going to understand what is reflection. Students, if I have a surface where a ray of light incident on it bounces back to the same medium, then it's called reflection. Here, the law of reflection says that angle of incidence is equal to angle of reflection. Therefore, students, first let me talk about plane mirrors. When you talk about plane mirrors, there are some properties. What are they? If you take a plane mirror, my dear students, I have a person or an object placed in front of the plane mirror, then we all know for a fact that object distance will be equal to image distance. Right? Height of the object will be equal to height of the image. These are some properties. And what type of image is formed, students? What type of image is formed in a plane mirror? What is the nature of the image, students? Very good. It is virtual. It is erect. Very good. And it is same size. These are the three properties: virtual, erect, and same size. Students, for entrance exam, very good. Dinesh, you are absolutely right. One of the other properties is it is laterally inverted, meaning this is something that we experience. If I lift my left hand in front of the mirror, it appears as if I have lifted my right hand. That is called as lateral inversion. Very true, students. These are all some of the features. Now, when it comes to MCQs, we need to be looking at moving objects in front of a mirror. Students, observe this very carefully. When I say that the mirror is a plane mirror and an object is moving with a velocity u, then image will also move towards the mirror with a velocity u. All right. So let's say they ask you a question. An object is moving with the speed of 10 meters per second with respect to mirror. Then what will be the speed of image? Students, answer will be minus 10 meters per second. Answer will be minus 10 because they are moving in opposite direction. But let me ask you one more question. What is the velocity of image with respect to object? What is the velocity of image with respect to object? Students, here we make use of relative velocity concept, where velocity of image with respect to object is velocity of image minus velocity of object. Velocity of image will be minus 10. Velocity of object is minus 10. So answer is minus 20 meters per second. Some of you have told 20. You're right. But 20 is speed. Students, 20 is speed. Whereas minus 20 is velocity. So if they ask you speed, you can go with 20. If they ask you velocity, you can go with minus 20. Or go with the most appropriate answer. At the same time, students, when I say that there is a mirror and an object is moving perpendicular to the mirror with a velocity u, then the image will also move with a velocity u. So if object is moving with 10 meters per second, my dear students, what is the velocity of image? That is also 10 meters per second. Then students, what is velocity of image with respect to object? What is the velocity of image with respect to object? That's beautiful. Velocity of image minus velocity of object, which is 10 minus 10, that makes it 0. Okay. Some of you had asked me to repeat the previous concept. I said that relative velocity is image with respect to object. Image is negative, object is positive. That's how we get what we see there out in the screen. Minus 10, minus 10, minus 20. Minus sign is used when they ask velocity. If they just ask speed, then we go with plus sign. Is it clear now, students? Those that ask for doubt, I hope it is clear. All right. Next one. I will extend this idea a little more. Observe. What if an object is moving with a velocity such that it makes an angle theta with respect to the mirror? Then students, we split the velocity component into two parts. The velocity along the perpendicular of the mirror is v cos theta. Therefore, this part will become minus v cos theta. Velocity along the horizontal will be v sin theta, right? Velocity along the horizontal will be v sin theta. Therefore, if they ask, what is the velocity of image? Then it will be v only, students. But if they ask, what is the velocity of image with respect to object? Then we split it into two parts. Along horizontal, we say it is minus 2v cos theta, correct? Along vertical, we say it is 0. Therefore, velocity of image with respect to object becomes minus 2v cos theta. Are we clear, students? Are we all clear, students? That is good. So these are some additional ideas required for entrance exams. Now, having discussed about motion of the plane mirror, students, next let me discuss about deviation. deviation. Students, if I have a plane mirror and a ray of light is incident at an angle theta, then it reflects also with an angle theta. What is the meaning of deviation? If this ray of light did not meet the mirror, it would have traveled straight. Because of the mirror, it has bent through an angle. So the deviation is nothing but the angle through which the light is bending. From the diagram, students, theta plus d is 180 degree. Am I right? Am I right, students? So from here, we get deviation produced by a plane mirror is 180 minus 2 theta. Please write this down. Deviation produced by a plane mirror is 180 minus 2 theta. Simple, na? So remember these formulas, students. Likewise, if I have two plane mirrors that are subtending an angle theta between them. If I have two plane mirrors, students, that is subtending an angle theta between them, then we say that any ray of light which incidents, it will go reflection twice, and this is how it will deviate. Once again, students, deviation is angle between incident ray and emergent ray. This is my deviation. Here, deviation is given by the formula 360 minus 2 theta. Students, 360 minus 2 theta. All right. One of you are asking how. I may not have time to prove it, students. It's a very lengthy process. I don't have sufficient time for it. If you require, you can always check it in out in YouTube. There are videos that shows how it is, but this is the relation. Okay, I do not have time to explain it. All right. So theta is the angle made. Second one. Then deviation produces 360 minus 2 theta. Now coming with number of images. For number of images, students, if I have an object which is placed symmetrically between two mirrors, then the number of images that are formed depends on the angle made by it. Students, if the two plane mirrors are making an angle theta, then number of images is given by 360 by theta, where if n is even number, then number of images will be n minus 1. But if n is odd number, then number of images will be n itself. I'll tell you why. Students, answer should always be odd number. That's why. Answer should be odd number. That's the idea. So I'll take an example. If theta is 90 degree, how many number of images will be formed? If theta is 90 degree, how many images will be formed? Okay, so one of you says 4. Observe, students, 360 by theta will give me 360 by 90, which is 4. But 4 is an even number or odd number? 4 is an even number. Therefore, number of images will be even number minus 1. Therefore, answer is 3. I hope you all understood the idea. Did you all understood the idea? One of you asked, sir, what if we get decimal number? Then you should round it off to the nearest integer. Decimal number means round it off to the nearest integer. Okay. So these are some other formulas that are connected to mirrors, my dear students. Number of images formed. So number of images formed, how do we find? First, you have to do 360 by theta. If it is odd number, leave it as it is. If it is even number, subtract it with 1. That will give you the number of images formed. All right. Students, with that, I have completed the theory required for plane mirrors. Let me next go to spherical mirrors. Students, in the case of spherical mirrors, spherical meaning it is part of a sphere. So let me say I have a mirror such that it is silvered from the outside. This is called a concave mirror. If I have a mirror that is silvered from the inside, then it is called a convex mirror. Students, this mirror is made out of or made from a sphere. It is a part of a sphere. The center of the sphere from which the mirror is made of is called center of curvature. The distance from the pole to the center of curvature is called as radius of curvature. If I take a narrow beam of light, which is parallel to each other, after hitting the mirror, they are going to reflect. They are going to reflect. Students, these reflected rays will never meet. Therefore, it appears to meet at a point. It's appeared to meet at a point. That point is called as focus. That point is called as focus. Students, we say focal length F will be equal to R by 2 for a plane mirror. Sorry, for a spherical mirror. For a spherical mirror, F is equal to R by 2. Is that clear? We have few more relations connecting mirrors. 1 by F is equal to 1 by V plus 1 by U in terms of object distance and image distance. One more, students. Magnification is height of image by height of object. Likewise, magnification can be also written as minus V by U. Students, if you remember some additional formulas, it will help you to solve a lot of MCQs, avoiding steps and in a very short method. Please take this down. Magnification is F by F minus U. And one more formula is magnification is F minus V by F. These two additional formulas will help you to solve a lot of questions from MCQs. So one of you are asking if it is 360 by theta, the previous one. All right, if it is 360 by theta and it's a fraction, then you find the decimal value, round it off to the nearest integer. Understood? You find the decimal value, round it off to the nearest integer. Okay? Is that clear? All right. I hope you have taken down these formulas, students, for your magnification as well as spherical mirror. Next, I would want to talk about some sign conventions. Students, we use Cartesian sign conventions. Listen to this very carefully. I'll give all the convention in one diagram. I'll give all the direct in one diagram. Students, let us imagine that the ray of light is coming from left to right. This is the direction of light. All right. Then, according to sign convention, any measurement made from the pole along the direction of light is positive. From pole away from the direction of light is negative. All measurements made above the principal axis is positive. All readings made below the principal axis is negative. So these are our sign convention, students. First, you need to identify direction of light, based on which you'll have to go with sign conventions. Having said that, students, if focal length is negative, what is the type of mirror? If focal length is negative, what type of mirror is it? Very good. It is concave. If focal length is positive, students, then it is convex. All right. Likewise, if magnification is positive, what can you say? If magnification is positive, what can you say? It is virtual and erect. Students, if magnification is negative, what can you say? It is real and inverted. Am I right? Real and inverted. Students, if the value of magnification is more than 1, then it is enlarged. If the value of magnification is less than 1, then it is diminished. Students, having said this, if I use a mirror such that I get magnification as -3, can you give me the properties? -3. Can you give me the properties? What are the properties, students? It is real, it is inverted, and it is enlarged, not diminished. It is enlarged. Why? Because I told you should take magnitude of M. Magnitude of M is 3, na? Understood, students? It is not diminished, it is enlarged. Please make your correction. Please make your correction. Understood or not? Students, if magnification is 2, then what is the properties? Magnification is 2. Virtual, erect, very good. Enlarged, beautiful. I am happy. Students, what happens if magnification is -0.5? -0.5. Real, very good. Inverted, very good. Diminished, perfect. Wonderful, student. So that's to do with your magnification sign convention. So this is all to do with sign conventions, my dear students. Having spoken about sign conventions of mirrors and also the nature of the image form, let me give you some key points here, students. First one, what happens to focal length if mirror is dipped in water? Will it increase or decrease? Students, can you tell me whether it will increase or decrease? Answer, students. No change. There is no change. What is the reason? What is the reason? We say F is equal to R by 2. So when I dip inside water, there is no dependence on refractive index. Therefore, it is independent of the medium. That is perfectly correct. It is independent of the medium. It is independent of medium. So there is no change. All right. Next one. I have a mirror. All right. And I cover half of the mirror with a black cloth. Half of the mirror with a black cloth. Then what happens to nature of image? Students, what happens to nature of image? Very good. Students, by covering, none of the properties changes. Size, shape, position, no change. No change in any of this. But what will change is intensity. So if I cover the half of the mirror, then the brightness of my image will reduce. All right. If I have the full mirror, it will look much more brighter. If it is half, it will look less bright. Students, this is the reason why when you go to these big parlors, there will be lot of mirrors around you because they want to appeal you by giving maximum intensity. But you will not get that same appeal when you are at home in front of your wardrobe, in front of your house mirror, because the mirror is small. The intensity of image that you get will be small. But when you go to a barber shop or a beauty parlor or a salon, you find yourself much more appealing because of the multiple refractions in size. So with increase in size, my dear students, intensity will increase. All right. So if one of them is covered, then intensity will decrease. All right. So these are some other ideas that are required for solving problems. My dear students, with this, I will get to MCQs. Shall we solve some MCQs, students? Good. Let us get to the MCQs. All right, first question. There are two plane mirrors. They are separated by a distance of 3 meters. A person is standing X meter to the right of the mirror and sees a series of images. Multiple images will be seen. Students, question is, what is the distance between, if the distance between the first image and the second image, the distance between first image and second image is 4 meters, then what is the value of X? What is the value of X? Okay. So many of you are telling 1 meter. Some of you are telling 3 meter and 0.5. See students, I will solve it for you. Observe very carefully. This is the plane mirror. All right. Observe my diagram carefully. So the person is standing at a distance X, which means the remaining distance from the second mirror will be 3 minus X. What is the reason? The total distance is 3. Students, let me say that this is the first image. This is the first image. And this image will be formed at a distance X. The first image becomes object for second image. The first image becomes object for second image. And therefore, students, for the second mirror, this becomes my new object distance, which is 3 plus X. Therefore, second image, my dear students, will be formed at 3 plus X. Second image will be formed at 3 plus X. And the distance between first image and second image in the question, if you notice, is given as 4 meters. Am I right? So therefore, students, 3 plus X plus 3 plus X will be equal to 4. Or we get 6 plus 2X to be equal to 4. I get X to be equal to minus 2. I don't have a negative option, but I have 2 as the option. That's why option 2 is right. Option 2 is the right one. That negative sign is because it is opposite to the first mirror. That's all. The negative sign is because it is opposite to the first mirror. X is 1, is it? Oh, I'm sorry. I'm sorry. My mistake. So I get 2x is equal to minus 2, x will be equal to minus 1. I'm sorry. My mistake. It is option 3. 1 meter. Is that clear to all of you? 1 meter is the answer. Okay, I'll explain it once more. For the first person, here x will be the distance. This will be x, right? The first image serves as the second object for the second image. Therefore, the distance from the mirror to this will serve as the object distance for the second image. If the distance is 3 plus x, then this will be again 3 plus x. That's how it is. So this way, students, we get x to be equal to minus 1. All right. In fact, you can look at it one more way also. All right. Let me tell you one more way. Let me show you one more way. Students, let me show you one more way. Let's say this is the first mirror, this is the second mirror. If I say that the first image is formed at this mirror, then we know this will be 3 minus x and this will also be 3 minus x, correct? Then 3 minus x, correct? Plus 3 will become the object distance for the second mirror. Therefore, image will be formed at 3 minus x plus 3. So image will be formed at 6 minus x. Correct? If it is taken from the other way. If you take it from the other way, students, 3 minus x plus 3 minus x. Sorry, 3 minus x plus 3. 6 minus x will be the object distance. Then image distance is 6 minus x. So 6 minus x plus 6 minus x will be equal to 4 meters. Correct? 6 plus 6 will give me 12. So I get 12 minus 2x to be equal to 4 meter. Right? So 12 minus 4 will give you 2x. 12 minus 4 is 8. X is equal to 4. Students, is this possible? The distance itself is 3 meter. Can x be 4 meter? Can x be 4 meter? That is why this method cannot be used. And our previous method is the only method possible. So this is not possible. Only the previous method is possible. Did you all understand, students? Okay. So I just give you another perspective. But that perspective turns out to be wrong. Next question, student. I think there's a simple question. Give me an answer. Very good. You're right, students. Option 3 is the correct answer, which is one of the properties of Cartesian coordinates or Cartesian sign convention. Next question, students. a plane mirror is placed horizontally on a level ground, and it is at a distance 60 meters from the foot of a tower. It is at a distance 60 meters from the foot of the tower. Just a minute, students. All right. So this distance here, students, is 60 meters. Now, read the next part of the question. The top of the tower and its image subtends an angle 90 degree at the eye. Students, after reflection, the ray of light will reflect like this. Therefore, the image will appear from opposite side. It will look inverted. And if you say that this is the eye, what the question is saying is the angle subtended to the top of the tower and to the bottom of the tower is 90 degrees. So this angle, students, is 90 degrees. We need to find the height of the tower. From the diagram, students, we know that if this is h, this will also be h, because height of the object will be equal to height of the image. Which means if I split this diagram into two parts, then this will be 90 degree. Because it is symmetric, students, can I say this angle here will be 45 degree? Because of symmetry, this angle will be 45 degree. Correct? Then we have a right angle triangle. Where the foot is at 60 meters, making an angle 45 degree. Then this is 90 degree. It's a right angled isosceles triangle. Therefore, this will be 60 meters. This will also be 60 meters. Correct? One more way to look at it, students. If you take it as triangle ABC, what is tan 45? Students, tan 45 will be BC by AB, where AB is 60. We know tan 45 is 1. So BC will become 60. Correct?
Marvin sir, I have a request. Marvin
Yes,
sir,
sir.
I have a
Yes, sir.
request.
Yes, sir.
Sir, Prajwal has come. He has come to the meeting. Can you actually make him co-host?
Yes, sir.
He's just below you.
Okay, yeah, there are two names.
Done, sir. Done.
Yeah.
Sir, you can
So
leave.
made both of them. Thank you, sir. Thanks,
Sir
sir.
can leave. Okay.
Sorry, sir. Yeah.
Sorry, sir. Sorry for disturbing you.
No problem, sir. So students, did we all understand how it is 60 meters? Yes. So the height of the tower will be 60 meters. Let's look into the next question, dear students. All right, so like there are many questions of this sort. There's actually a graphical question. Observe very carefully how I'm going to arrive at the conclusion. Students, first thing, they're telling it's a concave mirror. And for concave mirror, my dear students, focal length will be negative. If the real image is formed, students, if real image is formed in a concave mirror, then object distance will be negative. Correct? Image distance will also be negative. So from here, my dear students, object distance will be negative. Image distance will also be negative. Hence, if you use it in the formula, 1 by f is equal to 1 by v plus 1 by u. because all are negative, all will get cancelled. This turns out to be the equation for your mirror. Correct? Observe very carefully. What is plotted on y-axis, my dear students? It is 1 by v. Hence, I will keep 1 by v on the left hand side and bring 1 by u on the right hand side, so that I can equate it to the relation y is equal to mx plus c. Y is equal to mx plus c, where students quite clearly c is the intercept, which is a constant. C is the intercept, which is a constant. Observe one more thing, students. Because we have minus, slope will be negative. Students, slope is negative. Since y is equal to mx plus c represents a straight line, since y is equal to mx plus c represents a straight line, students, quite clearly we can eliminate option 3 and option 4, actually option 3, and because it is not supposed to be starting from the origin, we can eliminate option 2 and option 1, 3. Option 2 and 4 are eliminated. Option 2 and 4 are eliminated because they have positive slope. Option 3 is anyways not a straight line. That's when we can go with option 1, where we clearly notice that it has a positive intercept and a negative slope. Positive intercept and a negative slope. Are we all clear, students? Next question. This is a simple one. I think you should answer this. Think about it and then answer. Any of these is what you are telling students. Let us try one by one. See, if I use a plane mirror, all right, and I use a small lamp, what will happen, students, is the ray of light is going to reflect. Are the reflected rays parallel? Are they parallel, students? No. So plane mirror is ruled out. Let's try convex mirror. If I keep an object, again, rays of light are going to go away. So does it form a parallel beam? No. Students, if I use a concave mirror, then if lamp is kept at the focus, then the rays of light will emerge parallel, because rays which are coming parallel will meet at the focus. According to principle of reversibility, if object is kept at focus, image will be formed at infinity. Is that clear to all of you? Students, it is called as converging mirror. You are right, Ritesh. It is called as converging mirror. Are we all understanding? Wonderful. Next one, students. Megha asked me, what if object is kept at infinity? Students, I think we know when object is kept at infinity, image will be formed at focus. Correct? Image will be formed at focus for a concave mirror. Correct, Megha? All right. Next question. Students, whenever we get real object, please remember, always, remember this, whenever you get real object, U is always negative. U is always negative. All right. So from here, U is minus 20. Because it's a concave mirror, my dear students, focal length will be positive or negative. Focal length will be positive or negative. Students, because the concave mirror, it is negative, students. So focal length will be minus 10 centimeter. We have to find image distance. We have 1 by f is equal to 1 by v plus 1 by u. Please use the formula. Give me an answer quickly. It's a simple question. That's very good. Students, shall I give you a shortcut? Shall I give you a shortcut? Observe here. It's a concave mirror. All right. Focal length is 10 centimeter. Therefore, 2f will be 20 centimeter. We all know that when object is at 2f, image will also be at 2f. So answer is 20 centimeter without calculation. Did you understand the shortcut? Student, did you understand the shortcut? If focal length is 10 centimeter, 2f will be 20 centimeter. Therefore, when object is kept at 2f, image will be formed at 2f. One of you is asking, is it plus or minus? For a concave mirror, students, it forms a real image. Therefore, it is minus. Virtual image meaning it is plus. Understood or not? Students, let's say I got one more question like this. The focal length is 10 centimeter. I'll tell you how to use the shortcut in other cases. Focal length is 10 centimeter. An object is placed at 30 centimeter. Object is placed at 30 centimeter. Then where will the image form? Students, you should do option elimination. We certainly know for sure that when object is placed beyond 2f, when object is placed beyond 2f, image will be formed between f and 2f, which is between 10 and 20. So choose an option which is between 10 and 20, you will get the answer without solving. Without solving. Did you understand? Did you all understand? Good. So one of you asked me a question regarding the previous case. Previous case. That is for a plane mirror. Let me see if I can answer that. Plane mirror gives parallel beam of light is what Megha says, but not the fact. Students, if I have a plane mirror, the ray of light has to obey the law of reflection. Angle of incidence should be equal to angle of reflection. So observe here that the rays are diverging. They are not parallel. Megha, is it clear now? Good. Next question, students. A ray of light falls on a plane mirror. When the mirror is turned, this is a mirror, all right? When the mirror is turned about an axis, right angle to the plane mirror, meaning you are rotating it along x axis. You are rotating it along x axis. They are telling if incident beam is at 45 degree, then what is the angle between incident ray and the original reflected ray. So initially it was 45. After rotating, what will be the new angle is the question. My dear students, if I have a plane mirror, all right, and I rotate the plane mirror about an axis in this fashion, if I rotate the plane mirror like this, quite clearly, the normal will also shift, correct? Because this was my original normal. After rotating, my normal will shift. When normal is changing, students, angle of incidence and angle of reflection changes. But you observe here, because I'm rotating about an axis along x-axis, the normal is still the same. The normal is still the same, meaning there is no change in the angle of measurement. There is no change. If only it rotates such a way that normal rotates, only then angles will also change. Is that clear, students? Please make a note of this concept here. For other questions, students, if mirror rotates by theta, please write this down. If mirror rotates by theta, then image rotates by 2 theta. Then image rotates by 2 theta. Please write this down. This is one of the concepts that we use in MCQs. I will repeat this question once more. Plane mirror has an axis passing through perpendicular to the mirror, which is what you can see here, my dear students. If I rotate it about this axis, then the normal will not change. The normal will remain the same. But if I rotate it in this direction, then you notice the normal will change. When the normal changes, my dear students, it is only then the angles will change. But because in this question, the normal does not change, option 4 is the right answer. Is it clear, Arfa? Good. Next, students. Next question. Let us write down the ideas first. Let us write them down, students. When an object is placed at a distance 25 centimeter, as I told you before, take it as negative. The magnification is found to be M1. When the object is moved 15 centimeters away with respect to the earlier position. So students, if this is U1, then U2 will be 25 plus 15. That should make it minus 40 centimeters. Then the magnification, students, is M2. They're asking if M1 by M2 is equal to 4, what is focal length? Students, let us look at relation between M, U and F, because those are the three things that they're dealing with, M, U and F. What is the formula? What is the formula? Formula is M is equal to F by F minus U. Therefore, students, M1 will be F1 by F1 minus U1. Correct? M2 will be same focal length, same mirror. So let me not say F1, F2 and all. Let me simply say F. All right? So M2 will also be M2 will also be M2 will also be F by F minus U2. Students, take ratio, give me an answer quickly. Take ratio, give me an answer. All right. Many of you are telling it is option 4. Let me solve it. So M1 by M2, my dear students, will be 4. That will be equal to 1 by F minus U1 by 1 by F minus U2 because F will get cancelled. Therefore, it is F minus U2 by F minus U1. Applying sign convention, students, it is F minus 40, F minus 25. So students, 4 will be equal to F plus 40 by F plus 25. Cross multiply. 4F plus 100 will be equal to F plus 40. Or if you bring it to the other side, 4F minus F will be equal to 40 minus 100. 3F is equal to minus 60. F will be equal to minus 20. Triveni says, wouldn't it be F minus U1 by F minus U2? Triveni, you have not applied sign convention. Observe here, important, sign convention. Sign convention is important. Good. So answer is option 20, option 4. Next question, students. For the given diagram, the position of the final image after 3 successive reflections, taking the first reflection on concave mirror. Okay. First reflection on concave mirror. Students, let us do it one by one. First thing, when you get problems like this, image formed by the first reflection serves as the object for second reflection. Students, for the first object, we say U is minus 15, right? Radius of curvature is 20. Therefore, focal length will be minus 10 centimeter. Let's find image. 1 by F is equal to 1 by V plus 1 by U. Therefore, 1 by minus 10 is equal to 1 by V minus 1 by 15, or 1 by V will be equal to 1 by 15 minus 1 by 10. On taking LCM, I get minus 5 by 150 minus 5 by 150, or this will be 1 by V. So therefore, V will be, students, minus 150 by 5. Turns out to be minus 30. Turns out to be minus 30. Am I right, students? So observe here. If object is at 15, image will be formed at minus 30, meaning image will be here. Now, students, from the mirror, image is formed at minus 30, meaning it will be somewhere here, because total length they have given as 40. Can you see that? Total length they have given as 40. Total length from the mirror they have given as 40. Out of that, this is 30, meaning what will be this distance, students? How much is this distance? What is this distance, students? Very good. It is 10 centimeter. Students, if object is at 10 centimeter, can we say image will also be at 10 centimeter? Yes or no? For a plane mirror, object is at 10 centimeter, image will also be at 10 centimeter. So students, this is the new position. This is the new position after second reflection, because first reflection was from concave mirror, second reflection was from mirror. Third reflection, meaning this serves as the new object for the mirror here. So what is the new object distance, students? What is the new object distance? Anybody? What is the new object distance? Wonderful. It will be 50 centimeter. Wonderful. Because from mirror to mirror is 40. Again from here to image is 10. 40 plus 10, 50. Once again we use formula. 1 by f is equal to 1 by v plus 1 by u. Correct? Therefore, 1 by minus 10 will be equal to 1 by v minus 1 by 50 because it is negative again. On simplifying, students, 1 by v turns out to be 1 by 50 minus 1 by 10. Answer is 12.5. I'll have to simplify. I think some of them have got it as A, some of them have got it as B. Let us simplify. So you should get this as minus 40 by 500, or it is 1 by V. So therefore, students, V turns out to be minus 50 by 4, that is 12.5. So, Mr. Excited person, you are right. The one who got very excited. You are absolutely right. Did you all understand the method, students? Did you all understand the method? Mohan, did you understand now? Good, Mohan. Let us try the next question, students. A boy stands in front of a mirror at a distance 30 centimeter away from it. He sees an erect image and height is 1/5 of his real. So it is erect and diminished. Students, whenever we get an erect image, it is evident that it's a virtual image. But students, virtual image is formed in all the three cases. In plane mirror also virtual image is formed. In concave also it is formed. In convex also it is formed. But you know what is the difference? In plane mirror, it will be same size. In concave mirror, it will be enlarged. Please write this down. And in a convex mirror, it will be diminished. In a convex mirror, it will be diminished. So, because they are telling it is 1/5, it should be convex mirror. Please write this down, students. Please write this down, all of you. Is it clear, all of you students? Next question. I think this is a simple one. Please see if you can answer this. Okay, previous page, some of you are asking. There is nothing called plano-convex mirrors, dear. Plano-convex, plano-concave concept comes only in lenses. It doesn't come in mirror, okay? Afra, plano-convex, plano-concave concept doesn't come in mirror. It only comes in lenses. Okay. Dinesh, answer 3 is not true because virtual image for a concave mirror is always enlarged. Okay. Dinesh? Fine. Students, we'll discuss the next question. It's a simple one. See which is the right one. All right. Students, assertion says that the intensity of all the images are equal. Is it true? Not really. Not really, right? If you see yourself in a mirror, does all your images have same amount of brightness? No, right? So assertion is wrong. In case of parallel mirror, there are two images possible. Wrong. How many images? Infinity. How many images? Infinity. Therefore, assertion and reason both are false. Is that clear to all of you? Assertion and reason both are false. All right. So with that, students, we come to the end of this chapter. Let us meet in the next class with refraction. Thank you. You can take down my number, Karthik. You can take down my number.
Are you closing?
Yes, sir.
Yes, sir.
There are some students asking.
Okay, sir. Okay.
Yes. Yes, sir. I'm closing the sessions.
Yes, sir.